Welcome to the Barley, Malt, and Brewing Quality Lab. I'm Hannah Turner, Director of the lab here at Montana State University. We're super excited you're joining us for Craft Malt Week and to tell you about our breeding program, as well as do a tour through the lab and look at some of the projects that have us busy this fall. First thing, I'll hand you off to Jamie Sherman. She's down in our greenhouse and going to tell you a bit about how the breeding program supports craft malt.
I'm Jamie Sherman. I'm the barley breeder from Montana State University. And I wanted to give you an update about the breeding program in relation to the craft malt industry. So where we are is in a greenhouse in the Plant Growth Center. And this material that you see actually had its inception in the fall of last year. So let me give you a quick overview of the breeding process so you'll understand what's going on. So what we do is we select lines that are in some way different, and then we make a cross to make a new line, hopefully, that has the benefit of both of those parents. And the way we do that is by making crosses. And those crosses that made these families were made last fall. So we choose two parents and we make a cross. And when we make that cross, that first generation, the individuals have the genetic makeup from each of the two parents mixed in that new generation. And actually, all of those individuals are genetically identical because they're just, each one of them is half of one parent and half of the other parent. But then what we want is we want there to be genetic variation. So we want there to be this family created that has different composites of the parents. So we can get different sets of genes from each of the parents. And we do that through inbreeding. So we inbreed over 4 generations. We just let barley plants self like they normally do. And so then what we end up with are these groups of families that all come from one cross. So each one of these flats that's in this room comes from a single cross, but each line is genetically somewhat different from the rest of the individuals in that family. And so then what we can do is we can put them out in the field, we can do malt quality testing, and we can select from each family which line has the best composite of the two parents to give us what we want. What we've been looking for primarily for craft malt is to better control the speed of hydration that happens during the malt process. You know, we were really excited about our release, our new release Buzz, because it goes through that hydration process like 24 hours faster than some of the other lines that we've released. And so we're really excited about that because it could be a benefit to the craft maltsters. But another interesting thing that we found is Buzz also seems to have higher extract than a lot of lines that we've looked at. And so that gives us this power where you can vary the amount of modification that you do, and then that could end up impacting your flavor profile. So if you let it kind of over-modify, then you're going to get a lot more sugars and sweetness, and perhaps that would be good for a specialty malt, but then you can go through the malt process faster and get it modified just right, and then maybe that would be a really good, just a normal base malt. So we think that Buzz may give us some flexibilities, and we're wondering about that possibility with these lines too. So the speed of hydration we think is important, and we've really been working hard on that. Another thing that we've been working on is flavor, and we've done a whole bunch of work looking at old varieties that have been used over a number of years, and we've found that there are chemical differences. And now what we're really interested in is seeing if there's an interplay between those chemical differences and the level of modification the sort of malt process we go through, and then ultimately the flavor of that malt. And so to be able to do those sorts of studies and make them be genetic, you have to work on a genetic population. So we need these populations that are varying for these traits. For example, varying for the speed of hydration, or varying for different chemical compounds. And then we can figure out from that population what is the genetic control of that chemical component or the speed of hydration. And then we can then ultimately use that information to help us create a new line, and a line that has very specific attributes that would be an advantage to the industry. Now we're in the seed room at MSU, and the seed room shows us that even though the field season is over, we're not done with our work. So what you see is bags of seed that were harvested just a couple weeks ago. And now they're going through the processing step where they have to be cleaned, we get yield, we get plumps, we get test weight, and then they go over to the malt lab.
So it's late September here in Montana, and harvest has just been completed. All of our program, which encompasses about 13 people between Jamie Sherman, our breeder, myself here managing the lab, and we've also got another manager over on the seed side of the program, a few assistants, undergraduate, graduate, and PhD students. Everyone's all hands on deck getting things processed from harvest. The first thing that we do here, we run protein on it. So we've got our NIR here, which is going to evaluate both moisture and protein. And based off of those protein values, we'll make some final selections, make sure that we're comfortable with what's going to go into the malting process. And from there, we head to the malter. So here we are at our malters. We have CLP malters out of the UK. Incorporated in this, we've got 3 separate steep germ tanks. They're all identical. In these tanks, we load individual cages. These cages are able to hold 4 samples in each, and then 4 cages go per steep tank. So typically our malting scale is we load about 120 grams of barley into each one of these quadrants. That makes it so that we're able to malt 16 samples a batch, so close to 50 samples a week at that scale. We also have a method that really improves our ability to make selections for the breeding program in our early trials, and that's a pico scale, where we actually load about 6 grams of barley into one of these loose leaf T-balls. We can fit 6 of those per quadrant, and that gets us up to close to 300 samples a week that we're able to malt at that scale. So these tanks or cages just load down into the tanks. In the tank, we do both our steep and our germination phases. 2 days deep, we're able to fully control when the water comes in, what the temperature is. We're able to aerate with both moist and dry air. And then we've also got turning rollers in the bottom so that we're able to turn those cages and keep the malt evenly treated. So about 2 days in steeping and then about 4 days in our germination phase. So the cages cover the 2 scales, the pico at 6 grams and the micro at 120 grams. But we've also got a 3rd scale that we're able to do with these tanks. So we have these false bottoms that slip down right into the bottom. And then that allows us to malt up to about 8 kilograms per batch. And that's really been beneficial as we're moving into the brewing side of our program. It means that we're able to take barley all the way from the initial crosses through the full breeding process, and then some of our elite lines we're able to malt in these large scales and then actually brew with malt that we've brought up through our own process. So that's pretty exciting. So once the samples are finished in that 6 days roughly in the steep and germination phases, the last step is that the samples come over to our kiln. So in the kiln, the T-balls are actually loaded into a cage similar like this, and that cage is able to go directly into the kiln. Our 120 gram micro samples go into individual tubes and then are dried that way, and our large scale actually go into a kiln bin like this. And so that kiln, it very well mimics what a craft maltster's setup would be between temperature and then also controls. Typically for the breeding program, we're going to a lager style malt, so very low, slow drying. We're trying to preserve those enzymes, and that creates a good baseline for selection purposes in the breeding program. But now we're getting into brewing and we're playing around a lot more with what types of malt we're able to make. And so this kiln, beyond just doing certain temperature for a certain time and then kicking to the next phase, it can also control based off of differentials below and above the bed. So either a temperature differential or a moisture differential, and we can kick to next phases. And so that's made it so that we've been able to make lager malts, Viennas, Munichs, caramels, a lot of really good control and variation that we're able to get out of this kiln. So we're pretty happy with this system overall. So about a week that it takes for samples to go through this process. Now that we've got harvest coming in, these malters are booked up where we just have them constantly running. We're at a rare hour of the day where I've got one empty and I can show it to you, but we'll be filling this up a little bit later this afternoon, and it'll be about 8 months before we push through all of the breeding samples, and then we're able to do a little bit more experimental and playing around with some things beyond the breeding program.